During development of an organism, stem cells have the ability to give rise to multiple cell types in any organ. However, the mechanisms followed by the stem cells to give rise to multiple cell types is not completely known. I am interested to address this question as part of my PhD project, and we chose vomeronasal organ, also called as VNO, in mouse as a model system to study this question. In mouse VNO, there are majorly two different types of neurons. First one is type A neurons, and second one is type B neurons that I'm showing you in the PowerPoint slide as two different types of emojis. So when stem cells want to convert into different types of neurons, the first thing they try to do is to divide in numbers. So step one is to divide and increase in numbers, and step two is to then convert into different cell types. So in the beginning phase of my project, we were able to identify two different pools of dividing cells that come from stem cells. In the first pool of dividing cells that are about to become type A neurons, we identified a specific expression of a protein called as Notch ligand. Whereas in the second pool of dividing cells that are about to become type B neurons, we identified again a specific expression of a receptor called as Notch1 receptor. If you can imagine Notch ligand as a fist, and Notch1 receptor as a slot where it can easily fit. So there is a signal sending cell that has a Notch ligand and a signal receiving cell that has a slot where it can easily touch. So when these two ligand and receptor touch each other, the signal receiving cell now suddenly starts expressing more genes, because of which it will convert into a specific type of neuron. So this is what we theoretically came up to a concept. However, we have to experimentally prove that notch signaling is indeed is important for the formation of these two neurons. So we did two different types of experiments. In experiment number one, we removed the Notch1 receptor of a signal receiving cell, and when we did that, we saw more number of type A neurons forming from stem cells. On the other hand, when we artificially activated notch signaling in all the dividing cells, we saw formation of more type B neurons from the stem cells. So on the basis of these first and second experimental evidence, now we were able to say that notch signaling is important for formation of two different types of neurons from stem cells in mouse vomeronasal organ. Although I worked on only mouse vomeronasal organ, my study can also be applicable elsewhere in many organs in the body, because notch signaling is important for development of many organs. Thank you very much for attending my talk.